Welcome to the Explainer. So today we're taking what usually looks like a really quiet, maybe even boring laboratory process and turning it into what it actually is. A high-stakes, microscopic race where proteins are the competitors. We're calling this the molecular marathon. It's basically an incredible odyssey to separate and identify proteins. All right, let's head straight to the starting line.
Act 1. The Great Escape. Protein Extraction.
Now, right now, our tiny protein characters aren't anywhere near a starting line. Nope, they're deep inside an E. coli cell, completely tangled up and essentially held hostage inside a heavily guarded bacterial fortress. Obviously, we can't analyze what we can't reach, right? So our very first mission here is to literally bust down the doors. So, step 1, we hit that E. coli culture with the aggressive, high-frequency buzz of a sonicator wand. This sends these massive shock waves through the sample, just completely shattering the cell walls. But, that leaves us with a totally chaotic soup of tangled proteins and shattered cell pieces. We gotta clean that up. Enter step 2, the centrifuge. By spinning this whole mess at absolutely incredible speeds, we force all that heavy, useless cell trash to sink into a pellet mountain at the bottom of the tube. That allows us to swoop in and collect our glowing liquid prize right off the top, the soluble supernatant.
Act 2. The Equalizer. Sample Preparation.
Welcome to the sci-fi locker room. If we take a look at our newly liberated proteins, we quickly realize, well, this race isn't fair yet. These guys are all completely different shapes, and they carry entirely different electrical charges. To equalize the playing field, we send them into a molecular steam room for a process called denaturation. We unfold these crumpled, crazy 3D shapes into uniform, straight strings. We even use reducing agents to snip their complex structural bonds, kind of like cutting tiny pairs of yellow handcuffs. And now, enter our locker room enforcer, Agent SDS. SDS steps in and essentially sprays all the proteins down with a powerful chemical negative paint. It coats them entirely, so now, every single competitor becomes a uniform, straight stick carrying the exact same negative charge. Line up noodles, time to lose the curls. And just look at that number on the screen, 95 degrees Celsius. That is the intense heat required to literally melt these proteins down during this unfolding process. The heat breaks them down, and the SDS ensures they stay perfectly straight and negatively charged. Now, finally, our runners are equalized.
Act 3. Building the Obstacle Course. Gel Casting.
So we have to custom build our track, and it's really a tale of two gels. The top layer, right where they start, is the stacking gel. Think of this like a loose, open forest. It's got only a 4% acrylamide concentration and a pH of 6.8. This loose structure lets all the proteins loosely line up together at the starting line. But the bottom layer? That's the resolving gel. This is a dense, tight jungle made of 8% to 15% acrylamide at a pH of 8.8. The sheer density of this bottom jungle is what will truly, brutally test their speed.
Act 4. The Starting Gun. Running the gel.
Okay, our beautifully crafted gel track is set in the tank. The samples are loaded into the starting blocks at the top, and they sink perfectly into place thanks to these heavy glycerol lead boots we put on them. And flip the 120-volt switch. We've activated an electrical flow. Remember how our protein runners are coated in that negative SDS paint? Because of that, they are violently repelled by the negative pole at the top and pulled downward toward the positive pole at the bottom. That electrical pole is our starting gun. Here we go. The proteins race through that loose stacking gel and huddle together into a perfectly sharp starting line. Then, bam! They cross the border into the dense resolving jungle, and it's a frantic dash. The small proteins, let's call them our zippies, instantly slip right through the tiny gaps in the mesh. But the massive heavy proteins, our Samsons, they struggle immensely. They get totally tangled in the tight vines, slowing down to a crawl. The race just keeps going until the tracking die finally hits the floor of the tank.
Act 5. The Reveal. Visualization and Results.
So, the electricity is off and the race is officially over. But right now, we are staring at a completely blank, clear gel. Our champion runners are completely invisible to the naked eye. We have to develop the photo. So we soak the gel in a tub of Kumasi blue solution, followed by a destaining wash. Acting exactly like a chemical darkroom, the dye binds tightly to the proteins while washing out of the empty background. And there it is, finally revealing the crisp blue bands of our competitors. The official results are in. Our massive, bulky proteins are stuck way up near the top of the gel. They simply couldn't navigate the jungle.
Meanwhile, our tiny, agile champions are victorious all the way at the bottom. And the best part is, we know exactly how big they are, because the ladder squat ran right next to them. These are standard markers, acting like a ruler, basically holding up signs for 50, 100, and 150 kilodaltons, letting us measure their precise molecular weight.
This entire process takes invisible, chaotic cells and creates incredibly orderly, precise data. And it really leaves us with a fascinating thought about the profound real-world stakes of this laboratory technique. If identifying a single, specific blue band holds the secret to understanding a new virus, or maybe even curing a disease, what incredible breakthroughs might be waiting to be discovered in the very next molecular marathon?